going to look at your topical practice paper on photosynthesis. So let's take a look at it. Now, whenever you're doing your topical paper, whenever you're doing a question that's on photosynthesis, the first thing that you need to do is to write down the word equation. Same as for respiration. For these two topics, you must write down your word equation. So please pen it down somewhere. This will help you to actually do the rest of the paper. Okay? Now, write the names of the gases A and B involved in photosynthesis. So what happens here is that we know that the gas is taken in. Now, because there is sunlight, we know that photosynthesis is able to happen. So doing photosynthesis, okay, with the word equation. So I'm going to do the word equation first. Alright, so that we can then do the other questions. So we know that carbon dioxide together with water, as well as your light energy, in the presence of your chlorophyll. Now, just in case you all still don't know what is the difference, alright, the difference between chlorophyll and chloroplast. So chloroplast is basically a structure, okay, that is found inside your chloroplast. Okay. Now, so with these two together with water and carbon dioxide, what you actually will produce would be glucose. Now, some of you have written sugar. That's fine, because glucose is a type of sugar, and I will give it to you correct. But take note, once you reach secondary school, sugar is not accepted. You have to use glucose. Okay? So what happens is this glucose is what is needed for respiration. So you will find that the word equation for respiration and photosynthesis are the exact opposite, which is what I will show you later on towards the end of this tutorial. Okay? Now, it is also worthy to note that this glucose, alright, will be converted to starch, okay, when in excess. Now, this starch is then stored in your roots or at your fruits and everything else. Please pen down all these annotations. They are there to help you make sense of things and to connect the dots between one topic and another topic. Okay? So with this in mind, you will actually be able to answer A. So that's why actually I said to write down this equation right at the start, in fact. But since in this practice paper, the word equation is already part of a question, a second question in fact, I did not ask you to write it down. Okay? Now based on this, you would know that in order for photosynthesis to happen, you will need to take in carbon dioxide. So gas A, which is going into your tiny openings, these tiny openings that you see here, I'm going to color it, okay? This is known as your stomata. Okay, stomata for plural, stoma for singular. Okay, the whole itself is called a stomata, alright, or stoma. So gas A in this case would be carbon dioxide, and gas B would be oxygen, based on what you have written in the word equation. Now, be very careful. I also like to touch on this point that you will need to take note that respiration happens 24 hours, even for your plants. So if they were to tweak this question and the sunlight wasn't there, these two gases would be very different. So if sunlight was missing, then the gas that is taken in would be oxygen, and what is given out would be carbon dioxide. You realize that it's actually the opposite. Okay? Now, it's not that when the plant is photosynthesizing, the plant doesn't require oxygen to respire. No. Because the plant is already making oxygen, they will use the oxygen for respiration. And then they give out the excess. Okay? Now moving on to question 13. Anwar placed 2 setups below under sunlight for several hours. So keywords like sunlight will tell you that this is got to do with photosynthesis, this question. Alright? Now the airtight glass bell jar tells you that they do not want any gas to escape or come in. So there is a purpose for each variable to be stated there. Usually, as part of your answer, you will need to include this mention of these variables. Alright? And then they have plastic bags and whatnot. Now, Anwar measured the amount of carbon dioxide and oxygen in the bell jars at the end of the experiment. The table below shows the changes in amount of carbon dioxide in the 2 bell jars. Complete the table with the words increase, decrease, or remain the same. So over here, when you are doing an experiment, it's always important to identify your change and your measured variable. So this is your measured variable. This one is easier to identify because the word measure already is before these 2 items. And what is your change variable? So the change variable would be whether the plant is present or not, right? So you have your plant that is present in A but absent in B. So this is the change variable. Now you need to, why am I mentioning this is because as I marked on, I realized that there are students who would say the number of leaves, but it's not true. There is no leaves in B at all. So because of that, okay, your answer is wrong. Moving on. So the amount of carbon dioxide, okay, in jar A, let's take a look. So because jar A is able to photosynthesize, you would know that the amount of carbon dioxide should decrease. Now why decrease? Because your plant over here will actually take in your CO2. So because it is absorbing in your CO2, okay, the amount of CO2 will decrease. At the same time, when undergoing photosynthesis, excess oxygen will be given out. Okay? So therefore, your oxygen level will increase while your carbon dioxide levels will decrease. Now take note, some of you like to use chemical formula to actually craft your answer or to make annotations or whatever. Number 1, you are not allowed to use chemical formulas, alright, in exams for now at your age. If you insist on annotating with chemical formula, note that it's C, capital C, capital O, and the 2 needs to be subscript, meaning that it has to be at the bottom, just so you know. Same with oxygen, O2. Okay? Because the capital letter represents the different types of elements. So amount of carbon dioxide, as I mentioned earlier, would have decreased. Okay, and the amount of oxygen would have increased. Okay, for jar B, because there is no plant at all, there shouldn't be any changes, so it will remain the same. For both. Okay, now what was the aim of the experiment? So the aim of the experiment, as I mentioned earlier, wasn't to see how the number of leaves actually affected, okay, the rate of photosynthesis or the amount of oxygen or carbon dioxide, not about the number of leaves. It should be just whether, okay, or how the presence of the green plant affects the amount of carbon dioxide and oxygen in the bell jar. So many of you, instead of saying in the bell jar, you all tell me in the air. Okay, that's not right. So in the air, you mean here? Is that what you're talking about? In the air, outside surrounding, that's the air. No. So because you are specifically interested about the air inside the bell jar, you have to include that in your answer. So if you did not include that, please write down the whole correct answer. Okay? Now just to elaborate a little bit more, because at the beginning, you would have already identified your measured variable. So your measured variable in this case would be your carbon dioxide and oxygen. Now why is it not photosynthesis in this case? It is not photosynthesis in this case because there is nothing that you measure, okay, that you can relate it, confirm 100% related to photosynthesis. If this was going to be a question or the aim of the experiment is to look at photosynthesis, then you would be doing a starch test, right? Because you want to see whether or not starch has been produced. So you do a starch test, that's one. The other way is, unless it's a submerged plant, whereby you can see the number of bubbles being produced by the plant. These bubbles actually contains oxygen, alright? Okay. Next. At the beginning of the experiment, Arnold covered a leaf which was still attached to the plant with a piece of black paper on both sides as shown. He then tests the leaf for starch after 5 hours. So, there is a starch test. So remember, starch test, okay, you will actually is used to find out if photosynthesis has occurred. Okay? So using the leaf in figure 2, shade the portion of the leaf that will turn dark blue after he has removed the chlorophyll from it and adding iodine solution. So remember, this part is part of the starch test procedure. Okay, just in case you all forgot. Okay, the reason why you need to remove the chlorophyll is so that you would be able to see the color change clearly. So remember, chlorophyll is green in color. So if you were to stain the leaf, any color change is very hard to visualize if you don't remove the green first. Okay? Now, so this is the part that they add the black color paper. So think about it. What does the black color paper do? The black color paper essentially blocks out light. So because it blocks out light, it prevents this part of the leaf from receiving sunlight. If this part of the leaf cannot receive sunlight, then what will happen is that it's unable to photosynthesize. So brown iodine solution remains brown. Whereas this part over here, you would be able to see, okay, dark blue, because this part is where photosynthesis has taken place. So some of you have gotten this question wrong. Please take note of this. Okay? Next question. Now, this is a very, very common question. So in fact, without even reading it, you should more or less identify that, hey, this is actually a setup that is associated with photosynthesis. So what should come to your mind is that this setup can use to test many variables or many factors affecting rate of photosynthesis. So I'm just going to spend a little time talking about this just in case in exams there's many variation. So we know that for photosynthesis to happen, some of the variables will include light intensity as well as amount of carbon dioxide. So these two you can test using this setup, but you cannot test for water in this setup because the plant is already submerged in water. Just to recap with you. So to change the light intensity of the lamp, you can either place the lamp further away from the plant, or you can actually change the light bulb to one that gives a higher voltage or gives a higher watt or lux, LUX. Okay, that's one. Two, another factor that you can test for is how the amount of carbon dioxide affects rate of photosynthesis. Okay, so remember if they put there baking soda, this baking soda is used to provide carbon dioxide, CO2. So basically the more you put in, the more carbon dioxide, and that's how you can modify this experiment. Okay, we need to actually be aware because nowadays more and more questions will require students to think on their feet. Okay, so let's take a look. He measured the distance of the lamp from the beaker and the number of bubbles given up per minute by the hydrilla. He repeated the experiment 4 times by moving the torchlight further from the beaker 10 cm each time. He then recorded the results in the table below. So we need to translate this. So distance of lamp is actually talking about light intensity. So as my distance increases, I also need to know that my light intensity decreases as well. This is how you translate it. And please make sure that you use the correct description. It's actually light intensity. I don't want to see amount of light intensity. So from here, we'll be able to tell as light intensity decreases, number of bubbles also decreases. Now, this number of bubbles, okay, they contain oxygen. Okay, now this oxygen here refers to your photosynthesis. So obviously, the more you photosynthesize, the more bubbles you will produce. Okay, based on the above result, what is the relationship between number of gas or bubbles given out per minute and the distance? So, now this is the case that a lot of you have gotten it wrong. The most common mistake that I see is that your cause and your effect is swapped. Instead of saying as light intensity decreases, number of bubbles given out decreases, you swap it and say as number of bubbles decreases, distance of lamp or beaker decreases, or light intensity decreases. Okay, that is not correct. Okay, you should relate that it's due to the distance that results in the number of bubbles. Okay, so from here we know that distance actually represents light intensity. So because of that, you should talk about light intensity, no longer distance of lamp already. So you would say as light intensity decreases, the rate of, okay, the number, my apologies, sorry. This one over here, they're talking about this number of gas bubbles and distance specifically, sorry. So yes, so this one you have to follow strictly, okay? So it will be the longer the distance of the beaker from the torchlight, the fewer the number of gas bubbles given out per minute. Okay, so the cost needs to be at the front, and then the effect needs to be at the back, not the other way around. Okay, it's just like saying the more you eat, the heavier you get. This is correct. It cannot be the other way around. The heavier you get, the more you eat. Okay? Next, what conclusion could Mark make from the experiment? So because it's a conclusion, you need to see what is being translated. So distance of length is actually light intensity. Number of bubbles actually is talking about rate of photosynthesis. Okay, so you have to translate it. Okay, so what it means is that rate of photosynthesis increases with increasing light intensity. And one more other thing, when you talk about the distance of the beaker, you must have a point of reference from where to where. Don't just tell me the longer the distance of the beaker. From where? From here? From here? So because I need to have a point of reference, therefore you must tell me from where. Okay? Next. Okay, Oliver set up the experiment as shown below. He used two similar plants, both covered with a clear bell jar. Now, even words like that, you need to be sharp enough. Clear means what? Allow light to pass through. He coated all the leaves of B, okay, plant B, top side and underside with a layer of clear lacquer, like that of a paint. So this, we call this the change variable, because only one has it, the other doesn't, right? What would he observe on the inner surface of each clear bell jar after 1 day? Okay, I'm going to spend a little bit of time. Now, the inner side means this part. Okay, underside of it. Now, what he is likely to observe in these two experiments is that for A, you would actually see tiny little water droplets. Okay, I'm going to spend some time explaining to you why. Now, the hint here is a layer of clear lacquer. So, think about it. Why did they put the word clear? So, if you're sharp enough, like I told you earlier, clear tells you that sunlight can enter, okay, or light can enter. The leaves are able to get light, okay. So then ask yourself in a very strategic manner. So if it's not light they're inhibiting, what is it? Because some of you actually said that the plant could not absorb light. That's wrong, okay. So then after that, you ask yourself, is this really a question on photosynthesis? And the answer is Actually, no. Why? The reason why it's not on photosynthesis is because there is no what test? There is no starch test. There is no starch test or there's no bubbles. Remember I told you earlier, if it's a question that is on photosynthesis, you will actually need a starch test or at least have an underwater plant to count the bubbles. That didn't happen. Okay? So it's not. So then ask yourself, what was it that was affected? It was the leaves. So if you remember, the leaves have two functions. One is to photosynthesize and make food. Since it is not about photosynthesis, because there's no starch test, there is no plant underwater to count the bubbles, you would know that, maybe it's got to do with its second function, that is gaseous exchange. Right? So in terms of gaseous exchange, there are two things. Okay? There are two things that you need to know. So the two things that you need to know is, one, okay, it takes in oxygen for respiration at night. Two, it gives out carbon dioxide, okay, during respiration. So that's gaseous exchange. But there is one more other thing. It also allows water vapor to leave the plant. Okay? It allows water vapor to escape via the tiny openings known as stomata. Okay? So that is the source of your water vapor. Remember? So if there's tiny droplets on on this particular plant A, the jar of it, underside, that means condensation must have happened. So remember, in order for condensation to happen, there must have been a source of water vapor. And this is where your source of water vapor comes from, the plant. So this warmer water vapor, they will touch the cooler surface of your bell jar and lose heat to it and condense into tiny water droplets. Okay? That is why you will observe these water droplets over here. Now, let's talk about B. So because B has been applied with a clear lacquer, it blocks, it prevents your stomata from releasing or allowing the water vapor to escape, because you seal off all the tiny openings already. Okay, so if you were to go back up right at the top, so remember I told you the stomata. So because now a clear lacquer has covered it, okay, your water vapor cannot escape. Okay, now then after that, how do we craft this? Be very careful. Photosynthesis does not cause the water to become water vapor. Quite a number of you wrote this. You said because the leaves have been applied with a lacquer, hence photosynthesis cannot occur, alright, which results in no production or cannot produce water vapor. That is not correct. That is wrong. The water vapor comes from inside the leaves. Your leaves have water. It will change into water vapor as long as there's heat. Right? We all know about the water cycle. So where does this heat come from? There, the sun. So yes, there is still water vapor, but the water vapor cannot escape because the stomata all blocked already. Okay, for this one. So please check your answers. I put there a misconception if you tell me that photosynthesis causes, okay, water vapor to be formed. Okay, that's wrong. So how do we craft this? So we can say that water droplets can be found on the inner surface of jar A, but not for jar B. Okay, so we explain why. The leaves of B was coated with lacquer, which prevents water vapor from escaping from the stomata of plant B to condense into water droplets on the cooler inner surface of its bell jar. Okay, so somewhere you must tell me about condensation. Alright, if you can add in the point about it losing heat to the bell jar, even better. Okay, it's just that in this case, it's a 2-mark question. The key idea was for you to understand what the lacquer does to the leaf. Alright? Okay, moving on to the next question. Now, the 6 arrows in the diagram below shows exchange of gases between living things and their surroundings. So this is also another very common question where they showed you how respiration and photosynthesis works hand in hand to maintain the balance of carbon dioxide and oxygen on Earth. Because can you imagine if we just respire, okay, so remember these are, every single living thing needs to respire. So if we keep respiring, but we don't find a way to replace all the carbon dioxide with oxygen, we will eventually run out of oxygen. So thankfully, your plants, they are, through photosynthesis, they are able to take in the carbon dioxide and give out oxygen, okay, to replenish the oxygen that we've used up for respiration. So this whole entire diagram is very important. It shows you the relationship. Okay? So which 2 arrows represent the process of photosynthesis? So remember, in terms of photosynthesis, you will need to take in carbon dioxide. So if you take a look at this, my carbon dioxide is here and it's going into the plant. So straight away, I know that it's 3. And then on top of that, I know that something is given out, and in this case my oxygen is given out, the arrow is going out of my plant, so this is 4, 3 and 4. Now complete the following equation in the 2 blanks. Okay, this is the part that I want to stress to everybody. Please take note, you cannot and you must not put food. Food is the wrong answer. Okay, I stress again, food is not the correct answer, because obviously you cannot use a hamburger or a plate of pasta for respiration. You will need to eat it and you'll need to digest it. So yes, I will accept digested food. However, the golden standard, okay, is glucose together with oxygen. This is the golden standard. So you can tell me sugar, I will accept it, okay? However, please take note, golden standard is glucose. This is the most correct one, okay? So in the table, write P in the box besides each short phrase if it describes photosynthesis. Write R if the phrase describes respiration. So typically in photosynthesis, the word synthesis will tell you means to make something, right? So means to produce something. So food is manufacture is P, okay? Release from food, this is R. Respiration releases energy. Broken down, okay, that is also your respiration, okay, and then after that, stored as food is your photosynthesis, whereby excess glucose is stored as starch, as what I have mentioned earlier. Moving on, this part was very poorly done, so I'm going to go through this very quickly with you. It shouldn't be so poorly done, but it was really poorly done. So basically, they're asking for your word equation. So word equation will be carbon dioxide. Please do not use chemical formula. Together with water. Okay, and what you need would be light and your chlorophyll. And what you will produce will be oxygen as well as glucose. So remember, glucose is what is being produced in plant for photosynthesis. Glucose will also be the same thing that you require for respiration because they are opposite. So what are the factors that are required? Okay, so you will need carbon dioxide. You will also need water and light. Okay, so how do I know? I picked it up from here. And of course, if you want to say chlorophyll, you're also right. Just that we don't think of it, chlorophyll, as a factor because it's a structural thing that's already in the plant. Okay, the only way you can vary that is by adding more leaves. Okay, what does it produce? It produces, so like I always stress, take a look at your word equation. It produces oxygen and glucose. When does it occur? In the presence of light. So be very careful, those of you who put sunlight. I actually put a remark there. I cancel away the sun. And like I stressed to you many times, nowadays, okay, it doesn't mean that only sunlight can work. Artificial light will also work. And it's not the timing of the day. So if you tell me daytime, that's wrong. So respiration, what is the word equation? You will need to know that. Okay, so you remember it's the opposite. So in photosynthesis, you produce glucose. So in respiration, you will actually make use of glucose, okay, together with oxygen. So remember, you produce oxygen. So in this case, opposite. I told you that many times. Okay, and what you will give out will be energy. That is the most important thing, together with water and carbon dioxide. Okay, so some of you are thinking, wow, since respiration produces water, why do we still need to drink water? That's because the rate of water loss is actually greater than what is produced by respiration, at least for human beings. In certain deserts, there are certain organisms, because the water is so scarce, they literally just rely on water produced from respiration to so-called more or less survive. Okay, now what are the factors needed for this process to take place? So very straightforward, glucose and oxygen. What does it produce? Okay, it's also over here. Energy, water, and carbon dioxide. When does it occur? All the time. Okay, the minute you don't respire, that's it. Alright, you will no longer be alive. Alright.